Good morning, everybody. My name is Pratik Jagtap. I'm the session chair for the session called Microbiome Research in Core Facilities. And we have three speakers here, Kirsten Darcy from University of Vermont Cancer Center, who will be talking about metagenomics and metatranscriptomics. Gustavo Diaz from Colorado State University, and David Gaul from Georgia Tech. So Gustavo would be talking more about metaproteomics, how proteomics can be used for microbiome analysis, and David Gaul will give us an inputs or some insights into how metabolomics is used for microbiome research. We'll have 10-minute presentations from each of these speakers, and then it will be open for questions from the audience. But before we start, I'd like to ask Jeff Christiansen from QIAGEN, who's the sponsor for this session, to give us a presentation on QIAGEN products. Jeff.
All right, when am I using the fast forward here? Okay, awesome. Good morning. I see some familiar faces in the room, but for those of you who don't know me, my name is Jeff Christiansen, and I'm the sales director for the genomics, automation, and digital PCR teams at QIAGEN. And so today I want to talk to you a little bit about the value we try to bring to you guys as core labs and then to your end users. I'm going to do that by highlighting a couple different workflows that we have that I think will work for you guys. So that all worked. Some lawyer words. I'm a sales director, so I am very conflicted. That goes without saying. But if we jump into it, any of you guys who attended last year or attended any of the regional ABRF meetings, you probably had a chance to hear from Sam Ruley talk a little bit about what we were trying to do around some of our RNA chemistries. We are trying to give you guys as versatile tools as possible. We would love it if we could give you one kit that could replace 2 or 3 workflows that you have, right? Anything that can make your life simpler is what we're trying to accomplish. One of those products that fits that market is the QIAseq Fast Select RNA Library Kit, right? So it's a really simple kit to use. We provide you separate oligo-DTs and random hexamers, so you can decide, do I want to use this kit to do 3' sequencing and just get basic gene expression information really cheap and quick? Or do I want to use random hexamers and do full transcriptomic work? Or do I want to combine them both and get maybe even better transcriptomics on it? The other thing that we like to do is give you guys sample barcodes, right? And we do this right at the cDNA step, and this ensures a lot of that sample safety all the way through your workflows. The other kind of cool thing it does, especially if you're doing the 3' sequencing, is allows you to take a bunch of different libraries and pool them right after that cDNA step. So then all of a sudden, maybe you don't need a lot of automation to run a quick experiment, and it allows you to get some really fast answers. So the chemistry itself is quite quick. It's actually very automation friendly if you do have some liquid handlers laying around. It includes ribodepletion right up front. And at the very end of last year, we launched our QIAseq normalization beads. And these are beads that are designed to be used with pretty much any library going into an Illumina sequencer. And it gets rid of the idea that you have to do some dilutions and qPCR to figure out what you got concentration-wise or do a MiSeq run up front. This is a bead-based chemistry that allows you to do a really quick 30-minute protocol and replace some of those steps. And so it ends up giving you 4 nanomolar libraries at the end. And across the RNA libraries, if you look down there in the bottom right, I don't think you're going to find another way to get better equal representation across libraries with any other solution out there. So really quick thing, it works with our chemistry, obviously, but it also works with a lot of others. And so that's kind of a newer thing that's out there on the market. But that's just kind of the chemistry side, right? And if you guys know QIAGEN, you know that we also own CLC, and we also own Ingenuity Pathway Analysis, and so we spend a fair amount of time talking about bioinformatics as a company. And what we hear from a lot of your guys' end users is that there always is some kind of level of frustration, right? The amount of time it takes from them to go from getting a FASTQ file to getting the full bioinformatics workup to starting that next step in their experiment, to them is too long. And we understand that pressure. So what we wanted to do was make sure that we gave them a bioinformatics tool. This is not a bioinformatics tool for bioinformaticians, right? We have other pipelines for that. We have other products. This is a really, really simple tool that we provide with all of the RNA-seq chemistry you buy from us. It's free. It's something that pretty much anybody should be able to use, and it's something that you guys can pass on as a free value to your customer simply by using our chemistry. The way it works is you guys upload a FASTQ file. It does some initial alignment. All that uses a little token that came with our chemistry, and after that, you have a link that you can share back to the end user. They get access to the tool. They can pass that access off to anybody they want to, collaborator, somebody else in the lab. There's no further licensing that's required at that point. On the RNA side, it's a pretty simple tool, right? It all starts with that volcano plot in the middle. They are drag and drop lines. You can mess with the expression level and you can mess with the p-values and kind of narrow down to those genes that you actually care about. As you're doing that, obviously on the right-hand side, the heat map updates and it gives you a better idea of the clustering. The top left gives you a better idea of the differential gene expression and the statistics. And once they've kind of got it narrowed down to maybe the number of genes they want to work with or the genes they want to work with, they can kick on that bottom left-hand corner, and that's the Ingenuity pathway analysis. Now, this is a little bit of a light version of Ingenuity, if any of you guys are familiar with that. But it does give you canonical pathways. It gives you the upstream or downstream regulators. It gives you the upstream microRNAs that might control some of those things. And it also gives you an idea of the disease and the function that you're looking at, right? So anybody can walk up and use this tool. I've used it, and I am certainly not a bioinformatician, but you can use it to kind of narrow down the genes you're looking at, see how did the overall experiment work? What do I got to work with, and what are those next things that I might want to think about? And to help them further in that what comes next kind of conversation, that bottom right-hand corner is actually a link to GeneGlobe. And so whatever sorting and filtering they've actually done with their genes and gotten it down to their genes of interest, they push that button, takes them over to GeneGlobe with that same gene list, and they can select or deselect any of those that came over, and they can say, what other tools do I have to do orthogonal validation, right? Do I want to do a targeted NGS approach? Do I want to do some qPCR? Do I want to do some dPCR? All of that is right there ready to be configured. And so, you know, playing around with the tool for a day, ordering stuff the next day, within a couple weeks, they can be doing the next step of their experiment. And that's something that we hope brings value to them, and it's something that you get to offer them free just for using the chemistry. So it worked really well on the RNA side, right? We've had that out in the marketplace for about 6 months. We've had a lot of people try it out and give us really positive feedback on it, and a lot of folks implement it. So we wanted to do something similar to the whole genome space. And if you spend any time in microbiome community and you read some of those publications, it's probably not going to take you too long until you run into the QIAMP Power Fecal or Power Soil kits that are out there for extraction. These are really common kits. The other thing that you might find is the QIAseq FX DNA Library Kit. If you're not familiar with that, it's a protocol that we've had out there for quite a while, and it's a very standard one. That also pairs with those QIAseq normalization beads and can save you guys some labor and some time on your end. And then we also wanted to build a bioinformatics suite for those folks as well. And so obviously that's not the RAP tool, the RNA analysis portal, it's the MAP tool, and so it's the microbial analysis portal. But all of that's really built on that FX kit, right? And so if you haven't heard of that one, FX is our whole genome kit. It can handle any genome you throw at it, but it does incredibly well in microbiome. And so a lot of folks have used it, and it's a great one. It's a really fast protocol. Again, it's only about 2.5 hours. Only about 20 minutes of that is hands-on time. So if you don't have a lot of automation around, it's something you can do without too much pain and suffering. If you do have some automation around it, is probably our most scripted product that we have out there in the NGS space, right? So we don't have a big liquid handler, but we work with TCAN and Beckman and Hamilton and all those guys to make sure that our kits are out there and automated. This is also a kit that works really well with miniaturization. And so a lot of people have put this on their mosquitoes if that's something you're looking at. And just like the RNA chemistry, the QIAseq beads work incredibly well for these DNA libraries and, again, give you really good equal representation across all of it. But then just a quick kind of minute here on the microbial analysis portal. What does that look like? It's that same idea. You guys as the core director take the FASTQ file, you upload it, you name the samples, and then you've created the experiment. Once you guys have created the experiment, just like with the RAP tool, you can pass off this link to the end user. They get free access. They can share that access with whoever they want just by sharing the link. And again, any of the images you see from this or for the RNA analysis portable are completely exportable, and they're of a really high quality, so they're all ready for publications. On the microbial side, obviously, we give you ID in abundance of what's in each sample, right? And if anybody's looking at the antimicrobial resistance stuff, they also get that abundance in the profile for each of their samples. And the last thing that we provide is actually that differential abundance that they can kind of take a look at across their samples and across their experiments. So if any of that is if any of that's happening near you, right, a lot of times that next step will be either qPCR or dPCR. That's another thing that's near and dear to our heart. QIAcuity is our digital platform, and it is a very easy to use digital PCR system. It's a lot more similar to setting up a qPCR reaction than it is to doing like digital droplet and all those things that have been in the market for a long time. It's a much cheaper answer. So if any of that has any interest at all with you guys, I would encourage you to stop by the QIAGEN booth. If you walk on in, we're in the back right corner. Otherwise, reach out to a QIAGEN rep at any time. We love talking to customers, and I appreciate your time and hope you guys have a wonderful day.
Thank you, Jeff. We'll go on to Kirsten Darcy, who will be our first speaker, and she'll be talking about metagenomics and metatranscriptomics. Kirsten.
Hi, everyone. Thanks for joining us here on the last morning of this wonderful conference that we have, and thank you, Pratik, for the invitation to speak today. So we're going to be talking about the technical considerations for metagenomics. I work at a genome sequencing course, so we're really coming at this from the wet lab side of things today. Just a little bit about our facility. I'm with the Vermont Integrative Genomics Resource. I am the not-so-young Padawan to Scott Tye's Jedi Master of microbiome and metagenomics. I recognize some faces in this room, so I know people know who he is. We are a full-service center. We go from DNA extractions all the way through sequencing, and then we hand stuff off to our bioinformatics core. For sequencers, we have an Illumina MiSeq. We have the Singular G4 for our short reads. And then for long reads, we have a GridION as well as our brand new little P2 Solo. I'm not really going to go into how those technologies work. If you're interested in them, or PacBio or Element, they all have booths down at the vendor show. So one of the fun things with metagenomics and microbiome is it's not as easy as just taking cells out of culture and extracting DNA super easily, right? We think a lot about sample collection, preservation, how are we going to extract the DNA or RNA? And then we often do go back and forth on like, how are we going to synthesize the libraries? What's the best way to do that? Whether we're doing 16S or whole shotgun sequencing. And then finally, how do we want to do the sequencing? You know, do we want to do short read? Do we want to do long read? And we spend a lot of time with our investigators deciding what will be the best path forward for them and their project. So I've pulled a lot of slides from Scott. He's involved with the extreme microbiome. So a lot of this does apply to like more extreme stuff, but it does apply to, you know, taking soil samples. You're not just going to go out into a field, grab soil with your hand, stick it in a tube, and send it off to be analyzed, because you're introducing all sorts of contaminants that way. So, you know, for us, we like to consider from the very beginning, how are you going to reduce contamination and make sure that you're getting the best specimen that you can? So that can involve making sure that your collection tools are DNA-free or sterilized. Sometimes we have folks who are doing like wastewater studies, and so they have to go through and figure out what's the best way they're going to filter liters and liters and liters of fluids into something that we can use at the microliter level. So a lot of stuff goes into sample collection. Again, as far as once we get to DNA extraction, you know, that can be pretty variable. We test a lot of kits. This is a older study, I guess, at this point, almost 10 years or a little over 10 years. But, you know, methods can be variable, and you can get different yields depending on what you're doing and what the composition of your sample is. So do you have a lot of gram positives in that sample? Are there things besides just, you know, bacteria and archaea? Do you have fungi in there? We're dealing with this stuff all the time. What kind of matrix is in there? So we like to be able, if we can, or if the investigator can, take a look under a scope at the sample beforehand so that we have some idea of what the makeup is of what we're trying to extract. And then we can say, okay, yeah, we should use these enzymes, or we need to do a freeze thaw, or we're just going to beat the snot out of it with some beater beads and really get into it. We do have a couple of our favorite enzymes, so metapolyzyme for your gram-positives. We also have been using a lot of exopolyzyme for biofilms to really get through that. Those are pretty common, and cell walls are difficult to lyse. So, if we're looking at abundance of things, if you're not even getting into the cell to get the DNA out, that particular species will be underrepresented when we actually do the sequencing for you. So as far as methods go, the main ones for microbiome are kind of your classic 16S gene sequencing. We also do a lot of whole shotgun metagenomic sequencing, and as the cost of NGS has been decreasing, we've been seeing an increase in the amount of whole shotgun projects that we've been doing. We also have been dipping our toes more and more into metatranscriptomics to complement the metagenomic studies that our investigators have been doing. So briefly, since I think a lot of people in the room know how 16S works, you're basically just amplifying up these hypervariable regions. The most common, I would say, is V3, V4. If you have a different favorite, shout it out. We also have been seeing more and more full length because we do have Oxford nanopore technology. So that allows our users to get closer to species level. And then, so this is like a PCR amplification. For whole shotgun, you're fragmenting up the whole genome and sequencing that rather than just an amplicon of the genome. So some considerations when you're just trying to decide which method to use. Cost is a big one. Data depth, how far do you want to get? Do you want to get to genus, species? Are you interested in just like, I want to know what strains I have, right? Again, sample types, the composition of what's within your mix, and the analysis capabilities that your lab has or that your bioinformatics core has, right? So there are lots of pros and cons. I'm not going through every single one. Basically, 16S tends to be less expensive. Shotgun sequencing, the costs are coming down. 16S will usually get you to family, sometimes genus, depending on what you're looking at. Whereas with shotgun sequencing, again, you can get to species level. But it is more expensive, even though the costs are coming down. And with 16S, the data analysis tools are, I think, more readily available. Shotgun sequencing for that, the resources that are out there, the data that's going into them are sometimes a little spurious, but it's getting better and better and better. And one of the big cons, I think, for shotgun sequencing, and something that we focus on a lot, is host interference, right? So if you're doing like a gut microbiome study or something, you want to try to get rid of all the human or whatever the host is. Otherwise, you're just sequencing a ton of human DNA and not a whole lot of microbiome. So we said we have short read and long read. Originally, people were doing mostly short read, but the problem with that is that you often end up with a lot of gaps in your reads, especially if there's like high CG content within your microbiome sample. So usually, like in this example, it's less than 93% genome coverage, even though there was 140x sequencing depth for this particular sample. When you throw in long read sequencing, so this was done with Oxford nanopore, comparable sequencing depth, you're able to achieve 100% genome coverage because it is able, you're doing direct sequencing for one thing, and you are able to cover those more tricky locations within the genome that we often see with metagenomic samples. Moving on a little bit from genomics, we can also do transcriptomics. So this is on Oxford Nanopore, and you can do direct RNA-seq, which is actually really cool. There are a couple of special little things that we have to do because direct RNA-seq for Oxford Nanopore does require a poly-A tail. So we end up having to do a ribosomal reduction and then a polyadenylation of the RNA that's left, and then we can sequence from there. And you know, originally, like direct RNA with Oxford Nanopore was pretty low quality, but with their new chemistry, it's actually getting pretty nice. So just to wrap this up really quickly, you know, I think the take-home message for us, at least from the wet lab perspective for metagenomics, is that there's noise everywhere. And so we really try to hammer home bringing through controls for the kidome and for the extractome, and just trying to use DNA-free reagents wherever possible, starting from your collection. You know, we work with investigators, but we also have some of our own studies in-house, and you know, we have controls starting from collection, like this is our blank control, right? And we bring that control all the way through to sequencing. So I'd like to thank you for your attention today, and I think we're taking questions at the very end.
So moving on from metagenomics and metatranscriptomics, I'll ask Gustavo Diaz to give a talk on metaproteomics, so use of proteomics for microbiome research.
So good morning, everyone. I'm Gustavo Diaz, and today I'm going to speak about microbiome research in core facilities, in a specific case for metaproteomics. I'm a research scientist at the Analytical Resources Core at Colorado State University. So I'm going to go over briefly of our platform and workflow for metaproteomics. And then my idea is to present some challenges that we have faced while working with microbiome samples. So this is the platform that we use for peptide separation. We have this nano HPLC, the Vanquish. And then to work with very complex samples, we are implementing the use of FAMES. FAMES is a field asymmetric wavelength ion mobility device. It's a gas fractionation apparatus that we sit before the entrance of the mass spectrometer. In our case, it's an Orbitrap Eclipse. That's our team. I'm just going over for those of you that are not familiar with the metaproteomic workflow. This is a very general proteomics workflow. We have a sample, we extract the proteins, and then we digest the proteins into peptides. Peptides are separated using liquid chromatography, and they are infused in the mass spectrometer. And then we do a first round of analysis that we call the MS1, in which from intact peptides we collect mass over charge information and intensity. And then some of those, mostly based on intensity, are selected for a second round of analysis that we call MS2, in which the intact peptide is fragmented and the mass over charge of each fragment is collected along with the intensity. Then for data analysis, we use a FASTA file from a reference proteome. We do an in silico processing to get all theoretical MS1 and MS2 spectra. And then using algorithms, we do the matching of the MS2 to get information on the amino acid sequences. And also using the MS1 intensity as a proxy for protein quantity. And with that, we do the biological inference. For single organism proteomics and metaproteomics, those steps are very similar, but considering the very complex nature of metaproteomics, we need to do special considerations in each of the steps of this workflow. So before going any further, I just wanted to highlight the definition of the metaproteomics that was proposed 20 years ago is the large-scale characterization of the entire protein complement of environmental microbiota. And the main goal with metaproteomics is to obtain information, insights into the biological function of the microbiomes, and in this way provide orthogonal complement information for the other omics strategies. So here, as I told you, because of the complex nature of microbiome, we need to start just adding steps in each of the stages of the workflow, from the protein extraction to protein fractionation, down to building new set of bioinformatic tools for the data analysis. This figure here is a summary of the current methods that are used for metaproteomics studies, and is the result of a multi-laboratory collaboration. And I will just encourage you to take a look at this paper if you want to start working with metaproteomics. So in our specific case, I want to start just talking a little bit about sample processing. Because without a great extraction of proteins, no matter how powerful is your instrument, you will have not very good data. So in any sample, microbiome sample, gut microbiome or soil microbiome, you will have this very complex combination of cells. And as it was mentioned here, special attention to gram-positive and fungi, because of, the cell wall is very strong. So the extraction of proteins out of these cells needs to be carefully considered. The matrix effect also is important, and especially working with soil microbiome. We need to be mindful of the presence of humic compounds. Those can be up to 70% of the organic matter in a soil sample. And you see in the picture, SDS page of a protein with and without humic compounds. And the problem with those humic compounds is that they can be co-isolated with our process for protein isolation. And then they will interfere with the ionization of the sample, and then you don't have any good MS. So literature suggests that the use of SDS under high temperatures can help in removing humic compounds. And the figure there, the one in this one, is showing a result of our test using soil samples. And you see that the use of SDS in a relatively high concentration at 95 Celsius increases dramatically the number of IDs that we get. Thinking about gram-positive organisms, the use of bead beating seems to be very effective. This is, again, a soil sample, and we use a method with bead beating, intense bead beating, and the amount of peptides that were linked to gram-positive bacteria was a lot higher. And this is in line with this study, where the use of bead beating at different time points, 0, 1, 4, 9 minutes, increases the detection of actinobacteria, which are gram-positive. So then, moving a little bit to the right of the workflow, talking about fractionation. This is a very acceptable strategy when you are dealing with very complex samples. And I want to highlight the use of FAMES, because as I told you, FAMES is a gas fractionation device. And then in FAMES, by using different compensation voltages, you can select for different population of ions, as you may see here in these chromatograms. The cool thing with FAMES is that you don't need to do anything, any additional sample manipulation. You just select in your system different compensation voltages, and you will have subpopulation of ions that are analyzed at different moments. And additionally, FAMES will remove all the neutral ions and most of the single charge ions before they get into the mass spectrometer. So you will have a very clean front end of your instrument. But more importantly, when we use FAMES in a soil sample, we get a lot more protein IDs. So another challenge is the peptide spectrum match. The main thing is that normally we don't get metagenomic information of the samples under study. And that's critical, because without that, we need to rely on public repositories. And those public repositories generate a huge search space to do the in silico matching. And with that, we have a very high fault discovery rate scores that end up just filtering out high quality matches. So to deal with this, there is several strategies, most of them based on a two-step approach. And one of those was developed by our chair session, Dr. Pratik, and it's a very efficient way of dealing with the highest space and the fault discovery rate. And here what we do is like we interrogate the raw data against the huge data set without applying any fault discovery rate filtering. And then all the proteins that are identified, the FASTA file is extracted to build a smaller subset of proteins. And then this one is used for a definite fault discovery rate filter search. As you see here in the figure, this two-step approach was tested. And this is out of human microbiome raw data that was interrogated against 4 data sets. The first one is a huge database from metaproteomic information, above 6 million entries, and the second and third are just subset of that. It's still so big, you see 3 million and 2 million entries. And the fourth one is after applying the two-step approach developed by Pratik. And as you see, the number of identifications is much higher when you apply this two-step approach. In a similar fashion, different rationales, but still a two-step approach is this other strategy that we normally use. It's known as the happy ID. And in this case, you have your large data set. This one is over 9 million entries, 9 million proteins, and you select these HAP stands for highly abundant proteins, namely ribosomal proteins and elongation factors. So you get a subset of protein information, in this case, a little bit more than 100,000 entries. And then you do the search against this one to get something that they call the sample profiling. With this sample profiling, you have the more abundant taxa in your sample, and then you extract out of here the genomes of those highly abundant taxa, and you do a second search, a definite second search with a high fault discovery rate. So finally, one of the more complex challenges that we have is the taxa assignment and the assignment of biological function. And for that, we use the UniPet platform that was developed by a laboratory of Dr. Martens in Ghent University. And the taxa assignment using peptides problem is mostly linked to this concept of peptide degeneracy. That is, a peptide can be linked to multiple proteins. But in the specific case of metaproteomics, each of these proteins could be linked to different taxa. So you will never know which taxa assign the peptide that was the thing that you identified with proteomics. In the UniPet approach, they use the LCA, the lowest common ancestor approach, LCA as the most specific node in the tree of life that encompasses all taxa associated with the matched proteins. And in that way, you end up with a one LCA for one peptide, which is cool. And finally, for the peptide function assignment, which is one of the most important gains with metaproteomics, we have the same problem. We have our peptide of interest that was identified with our proteomics, but the peptide can be linked to several proteins. And at the same time, each protein may have one or more biological functions. So the biological function is inherited in the peptide by the protein of origin. So you end up with multiple biological functions for each peptide. But in this case, you don't have any LCA type approach to do a comprehensive biological function that encompasses all of the biological functions associated to your peptide. So the UniPet strategy is to do a weighted biological function score based on the number of proteins that are assigned to your peptide. So I will leave it there, and I will be here for questions. Thank you.
Thank you, Gustavo. I think we'll have the last speaker now, David Gaul, who will be talking about metabolomics in microbiome research, and after that, we'll open it up to questions for all the 3 speakers.
Thanks, Pratik, and the organizers for and inviting me to come give you a little bit of an insight into at least how our core operates and deals with the incoming projects in the microbiome space. First, I just want to introduce my team and a little bit of our facility. We started just about 10 years ago in about 2015. And I started with only one instrument on the metabolomics side, and on the proteomics side, we had 1.25 instruments. And from there, we now are up to over 10 instruments, and I had showed 3 buildings here because I'm spread out over campus in 3 different facilities. It has its pluses and minuses by all means, and we're also, but we've been able to grow steadily. I think now we have lots of instruments. Now we're looking for more people to help run all these instruments. One of the things that we try to do, and maybe a little bit different from how some other cores may operate, the term core sometimes is a little bit confusing in that a lot of people think, oh, it's a place where users can come in and utilize and use these equipment and perform their research. We are really more of a service core, and why that is mainly because the volume of samples that we do and the types of studies that we do. If we had a lot of all these instruments that were down, we would never be able to do any type of research for everybody. So one of the ways that we try to do it with such a small staff is we have to kind of keep control over the instruments themselves. This doesn't mean that the researchers don't get to come in and work with us. No, we all by all means invite them in and they can sit there and do everything. They just can't push the button to put something really nasty down our columns or something that's going to take it down for a week. So, that's one of the challenges that we do, but it's really not too bad overall. In terms of metabolomics, this is, for anybody who's in this field, this is a very generic slide, but this is mainly just telling the general workflow through any design experiment, whether it be microbiome or not. And the purpose of me putting up this slide was more to say that we interact in this full scale. We're a full service community, a service center where we can start from sample prep, really before that, experimental design. When I first started, we were lucky to always be in sample prep. And it took a few years to be invited to the table to be discussing the experimental design so that we can help reduce all the technical variations that were unwanted. And we go through, hopefully, all the way to pathway analysis. But it would be great if that was, here's a box, you can check on it, here's the price, and this is what you're going to get for it. But unfortunately, that bottom 2 boxes, identification or annotation, and the biological, those are things that are not easily definable, depending on, especially in a discovery type of experiment, non-targeted. Those could take anywhere from a couple hours to a couple years. And of course, as a service center, that's a very hard thing to try to quote anyone. So really, I get to the point where we can have some basic statistical analysis, give them direction, and then we can handle all the rest from a different standpoint. So from my perspective, my defined box is we can do anything from experimental design up to that basic analysis, and that's what you pay for. And then everything else we have to deal with on another way. In terms of microbe analysis, and kind of falls along the same lines, we want to take a complex sample, and these are just as complex as anything else that comes my way. We need to find some way to extract the analytes of interest. We do usually most of the heavy workload in my particular core is in LCMS, but we have GCMS, we have MALDI, we have DESI, we have lots of other tools that we employ in all different situations, but I'll say for the most part, LCMS is by far the most dominant technique, but it also is the technique that I have the most tools to attack that identification annotation problem. So that also plays a big role, because of course, once we have all these, we can detect lots of things. We can see potentially lots of fold changes, but what really comes down to it is you really want to know what that molecule is, and then was it really changing? And then hopefully then we can paste that onto a pathway of some sort, or you use of your collaborator to give the biological story, which hopefully will make a very nice paper at the very end. And if we can help get through to that end goal, that's our drive. That's why everybody on my team does what they do. I just mentioned the LCMS. This was just to give you the full breadth of metabolomics tools that are out there. I'm not going to go through every single one of them, but you can see there's a whole different variety, but really it comes down to 2 basic things. One, it's, even though we have a lot of either shotgun type techniques or direct infusion, we can do all of that, but we work with such complex samples that there's too many compounds to visualize at one moment. We need separation. So therefore, we can employ again, in my lab, it's mostly liquid chromatography. We have gas chromatography. We have a little bit of capillary electrophoresis. There's more techniques, but we need to have some separation so that we can have a better way to detect a lot of these molecules that we're interested in. And of course, in the mass spec, we have a lot of different ways to ionize. It can be electrospray, again, is the most common. We also have the ability to do atmospheric pressure chemical ionization, APCI. We can use inductively coupled plasma. We can use MALDI. We can use DESI. All of those techniques are just because we ultimately have to get everything into the gas phase and be a charged species to be able to do anything with it. So, but there's other complementary techniques. I do work with a lot of colleagues that do NMR studies. We try to do complementary type things. There are certain molecules that the NMR sees a lot better and they know what it is, as opposed to, and one example would be glucose. I can only tell you that it's a hexose. I can't tell you that it's specifically glucose because the tools that we have do not distinguish those isomers or separate those isomers. But the nice thing about NMR is they have the ability to do that. But we could also use UV, IR, fluorescence, which a lot of our microscopy colleagues use very often. But again, for our purposes, we generate hundreds to thousands of features. A feature, for all intents and purposes, anybody who's not a mass spec person, is just simply a peak. We don't really know too much about it. It just has an M over Z, and it has a retention time. So we call it a feature. And why we call it a feature is that there could be thousands of peaks, but every compound may not only be one peak. It could be multiple peaks. So we have a lot of tools that try to help simplify this down so that we are starting to get to the point when we go into that biological pathways, everybody wants to get down to one compound, identification of one thing going forward. In the experimental design part, this is where we get a little bit of fun, but also this is where, when we interact with customers, is the biggest pain point because there's only but so much money in one particular project. And of course, when they first come to me, they always have the comment of, I want to see everything. Okay, that would be great. I want to see everything too, but we're going to have to make some choices. I can tell you when I first started, very rarely I would have the conversation, is there anything of interest in the entire metabolome, entire biological world that is of interest to this project? And the answer usually is, no, I don't know yet. That's why I'm doing this study. And that's okay. I'm okay with that because at the end of those experiments, I'm hoping that their goal is to get a direction. I don't want them to have a complete answer because the nature of our measurements at that stage is pretty precursory. They can get a lot of information out of it, but it's not the end final thing. They're going to have to do another targeted experiment to make sure that they verify what they find. But with that being said, yeah, if everybody has plenty of money, we'd love to do a nonpolar and a polar extract. We'd run both in positive and negative mode, but you can start to see this is already 4 assays, and based on time and money, that's already pretty expensive for per sample basis. Then as we start to get into more specific things, it's a good and bad thing. When people started coming with specific targets, it's great for me because then I can start to direct them to certain assays that would be more beneficial to them. But then we also we start to learn that our assays are not always the best for when they tell me multiple things. Because it's always great to target one thing. We can do that very, very well. But as soon as you ask me to do 2 things, then I have to have a compromise between the 2. And that's sometimes a big challenge, but that's what we're here to develop the methods for, to come up with the best possible solution to hopefully obtain the information that you need at the time. Just want to kind of brief you on a couple of the studies that we've done, especially in the microbiome. I am part of a Molecular Transducers for Physical Activity Consortium. This is an NIH-funded consortium that was looking at, for the basic purposes, everybody probably in this room can appreciate that exercise is good for you. Well, our purpose was to try to figure out some molecular reasons why that is actually the case. So we started about 6 years ago. We had both planned animal studies as well as human studies. What I'm showing here is what will be published in early May, is one of our, the training study for our rat cohort. This basically, we had a series of rats, and we had a great team that went and dissected these. It was a multifaceted team that broke up into 3 areas. They were able to extract 20 different tissues out of these rats in a very short time frame. I truly am impressed every time that I hear people talk about it, because I've been crucially involved with harvesting organs here or there, but that's only one. I couldn't imagine trying to coordinate 20 for a large study of 60 plus rats. It was quite a feat. But in this particular study, we had sedentary animals. We had animals that were trained for a week, 2 weeks, 4 weeks, 8 weeks. And that training, they basically are running on a treadmill, and they're measuring their VO2 max. And as they meet their goals of their exercise, then the ramp in which they're running and the speed is adjusted so that they maintain a constant load on each of the rats. And after that, they were sacrificed, and then we were able to look at all the tissues. Again, the tissues, that's what's going to be published. What I'm showing you here is some just preliminary information that we came back with a lot of ancillary studies. And one of the studies is we were focusing in on the liver. And one of the collaborators, what they wanted to look at was they did collect fecal samples. So we did this work. So I'm just showing you briefly the total ion chromatogram for these fecal pellets from these rats. And one of the things that I was a little amazed at is just how rich they were in TGs. This is a total ion chromatogram in the positive mode, and all the features that you see kind of more on the right-hand side of the spectrum, those are all triglycerides for the most part. There's some cholesterol esters in there, but the predominance of how many were there truly was amazing to me. The score plot, the PCA score plot, I know you can't see the legend very well at the bottom. One of the things that we did notice quite a bit in a lot of the rats were that there was a sex difference in a lot of the organs, as one would expect in some cases, but maybe not in others. But we also noticed that there was a good, the separation here between the orange that are male rats and the female or in blue was also preserved there in the fecal matter as well. One of the other directions that we're trying to move into, and this is what this particular slide is capturing, is again, this is brand new data that I just received earlier this week. One of the focuses of a lot of researchers, and it's probably the hottest topic moving forward, is we want to get down to single cell metabolomics, just like our colleagues in genomics and proteomics. They're gotten a little bit further along, but we want to be able to do the same thing. And it really comes down to being able to work with the sample and have enough sensitivity to be able to detect the samples. We've already tried, I can tell you, a couple different ways that weren't very fruitful. This most recent one was a collaboration with Thermo Fisher Scientific. Alex and Fernandez's group, she grew up some cells, and we gave them specific concentrations of cell extracts. The 3 that I'm showing you peaks for, and I just selected one peak. This was not even this was me going in manually to go find something to get me really excited about this, and I found a cholesterol ester. This particular one is a cholesterol ester with a 16O chain, and what I'm showing in the bottom part of that slide is just the MSMS that helps confirm that it is indeed that molecule. The 3 peaks that you see, the top one is about 100 cells, then it's 10 cells, and then it's 1 cell. And I put a little graph to show that there was somewhat of a linear relationship to that peak area and the number of cells. I'm not going to say that it's exactly 1 cell. I can't really guarantee that. Hopefully, we're going to be doing more experiments in the future to help make sure that it's 1 cell. These are based on dilution, so they're somewhere in that ballpark. But so far, this was done with a Vanquish Neo, so we're a little bit lower flow than a normal, with normal flow, but we're not down to the nanoflow. For anybody who's worked with nanoflow, working with lipids and nanoflow is not easy for students to work with, so that's why we wanted to go a little bit higher flow, and we've been using the Neo. And this was collected on Explorers 240. Right now, I mean, we're going to be diving into this data set a little bit more, but I'm already very excited to see that we're going to be able to do quite a number of things, I think, on the single cell level, but it's going to take us also developing a few new tools to be able to do this really, really well. Overall, I mean, if you guys are partially in this space, then I don't think anything that I'm going to say in terms of future directions are going to surprise you too much. We constantly will always need improvements in how we separate and detect molecules, or metabolites in particular. And I think this is where it's highlighted most in the development to analyze single cells. I say single cells, but this also could be single bacteria. If we could get down to that level, that's going to help deconvolute a lot of our complex issues with complex samples. Pretty much anybody who's done metabolomics, the bane of our most of our studies is the fact that we need better tools to do annotations. And I can say that from my perspective, we have tons of tools that are out there. None of them take the place of a human. It still requires us to go back through and curate the data to make sure that we're giving reliable data back to our customers. I wish that wasn't the case, but I understand what the problems are, but these are generic algorithms that are applied to complex situations, and they don't always work perfectly. So taking away the human factor is, at least for right now, is not going to happen, but we have to find ways to help make this go a little bit faster. I think another couple areas where, specifically for microbiome research, is we definitely need more data repositories for microbial compounds so that we can capture what compounds are from each microbe, but also their functions, as well as any type of response to whatever the researchers may be doing in terms of a permutation to that system. If we start building these databases and these repositories, it's going to be a little bit easier to be able to look at new data sets and apply that to what has already been known and try to figure out where we stand and where we need to go. And overall, I think all my colleagues would probably agree that one of the things that we're really lacking is in terms of multi-omic data sets. We don't have a simple computer program that would help us bring all that together. It's still a very manual process. I work with spreadsheets. We can do correlations. We can do all the tasks, but we don't have a really good framework in order to do that type of research and do it quickly. I should mention the motor pack. I focused just on the metabolomics, but really that's a multi-omic process. So we do have genomics all the way through metabolomics, transcriptomics, everything. It's a huge data set, and the data scientists that work on it, they work on the entire thing. And I can even tell you from that standpoint, they're building their tools as they go. And there's no consistent tool across the board. So that is about all I have. Thank you.
So with that, we'll open to questions. Any questions for any of the three speakers that you have? Anybody? Okay, I have a question for all three of you, and maybe we can go, starting with Kirsten. In terms of the range of, projects that you get for microbiome analysis, what's the scope of data sets do you get, like in terms of how large they are as compared to some people who just come to explore some of the microbiome properties?
Sure, yeah. We get a wide variety of projects, right? Like I mentioned, we've got bioreactor guy, we've got, we at University of Vermont, we're pretty ag-based, so we end up with a lot of livestock projects, actually. And sometimes it's just like hey, we're interested in staph, but we need to know which strains are in here, particularly with milk production. And then, sometimes we end up just getting a couple of samples from somebody, or we also have projects where people are sending us, 3 96-well plates to process. So
Yeah, and then how much time do you spend in standardization for large projects before you get into the actually working on the samples?
It depends on the complexity of the samples, really, right? If somebody's pulling something off of a cave wall and this is the only sample they have, then we spend more time being like, okay, we got to get this right because they can't go back to whatever sulfuric cave they pulled that off a wall of. Whereas with the, like this guy's growing something up in a bioreactor, he can grow more, or he has a large amount of sample that we can go back to if something happens, right?
Yeah. Okay. Gustavo, same question for you. Like, what's the range of complexity of projects that you get?
Yeah, we work a lot with soil microbiome, and we are lucky because we have the metagenomic data for that one. But we have, for example, a group of researchers interested in the microbiome of air. Those are very difficult samples to work with. In most of those cases, it's difficult for them to get the samples, but then we use the samples just as a trial and error approaches in order to, standardize how we are going to get these proteins out of these, how we are going to separate these. And we have seen, for example, with the air microbiome with FAMES, for example, we have a bunch of, crappy spectrometry. When we apply the FAMES, the files went from 2 gigabytes size to a little bit of, let's say, 100 megabytes. So most of the information was filtered out as not being, peptide associated. And we also work with some gut microbiome studies, human gut microbiome. And those are relatively great because there are nowadays we have a big number of data repositories out of metaproteomics data, metagenomics data for gut microbiome. Yeah, but that's pretty much it.
Yeah. And how much do you rely on a good annotated, assembled metagenome data as compared to public repositories? Because you seem to use the high ref method, but also
yeah, with the soil, for example, if we compare the two approaches, let's get from the from UniProt, all the panproteomes of prokaryotes, and let's try to do the two-step approach versus having metagenome data. We get from metagenomic data by using FAMES in soil samples about 7,000 proteins compared to 3,000 to 5,000 using two-step approach. Yeah. It's still, I gotta say, the number of PSMs, peptide spectrum matches, compared to the entire data acquired, it's really low. I mean, 10, 15%.
Okay. So that's for the metagenome-derived database, or?
With metagenome-derived data, it got up to 30% in the best case scenario.
Okay. Also, in terms of quantitation, I mean, you mentioned MS1 that you use. Have you explored data independent acquisition method, ZI, or not started?
We haven't. We were very interested in doing that because we know data completeness is going to be a lot better, especially for statistical analysis. Yeah, but so far we are just doing DDA.
Yeah. And David, coming to you in terms of course, the complexity of samples, but also, when a researcher comes to you, when do you decide, or how do you decide that you're going to take a discovery approach versus a targeted approach, or do you always prefer a discovery approach and then go to targeted approach?
So I would say overall, the projects that come in, we have a wide variety. A good number of them are usually, they would like to get away with a lot fewer samples than what they probably should. It's especially in the microbiome projects, they usually haven't been too sample limited, which has been nice. They usually have enough money for enough animal studies, and they're never sample limited, which is good. If we had to go to sample limited type projects, then that's where a lot more give and takes have to happen. We try to do whatever we can to get the most out of one sample extraction, plus maximize the information that we'll get out of one particular injection. When they first come in, I do like to discuss the project and try to get a insight on what their goals are. Is this for a paper? Is this for a grant? How far along are they? What are those goals? And then that helps dictate what, should it be a more targeted or what we kind of maybe incorrectly call semi-targeted approach. The nice thing with a lot of our machines now is what we can actually do is that we can kind of do both. Within a particular LC run, they could tell us that we're interested in targeting one particular molecule, but we have a lot of other wasted time. We might as well just collect it. So we also collect untargeted around the targeted, and we can do both that way. I mean, the only downside of that is wherever that targeted molecule came out, we have a hole in the non-targeted portion. But that also generates a lot more data, and whether or not they have the resources in order to see that through to their end goal. In terms of the microbiome, I think we've seen both fecal, I've done tissue into the gut tissue of both mouse and human. We've done coral microbiome, and most of the questions are comparative to some of the other projects that we have, are still on the more general side, I would say, across the board. I don't know if other people have other things that they've seen or observed, but it's more of a direction. I would say that probably at the very beginning, when the very first one came to me, they were under the impression that metabolomics would be able to give them all the bacteria species that were in their sample, and I had to tell them that I didn't have the databases to be able to have enough specific molecules to determine what. And then it took me a little while, and eventually I figured out, oh, they had read all the papers on the genomics and the proteomics that actually could do that. And yeah, they just thought that, oh, naturally all the small molecules must have that same ability. But unfortunately, we need that knowledge base on what are the specific molecules that each of them are highly specific to, that we can then address that. But one of the points that I'd bring up is that one of the things, we have a constant changing matrix. Matrix is the background. And as from sample to sample, if the bacterial colonies or the microbial colonies start to fluctuate, we're starting to change that background. So having that knowledge from the other omes to know the approximate amounts will only make our job in data analysis of our data a lot easier. It's not going to make it simple, but it'll make it a lot easier, because then I'll know what the contribution of each one of those particular species are, and then we might be able to adjust the analysis based off of that.
Okay, thanks. Any questions from the audience?
So in the sort of extracellular space and the way that microbes signal each other, or even hosts signal their component microbiome, have any of your techniques been used to sort of look at what's going on outside the cells? I suppose proteins would be the obvious thing, but maybe metabolites and maybe even nucleic acid.
Here, I'll jump in. Yes, we have lots of studies that will look at the supernatant around in the fluid, and that gives a whole host of things. Now it's, unfortunately, in that extracellular space, to me, is a lot more complex, because then I can't tie it to any particular thing. Now, granted, the fecal matter is probably the same way. You can't tie it to any one thing. It becomes hard. But I think that's where I love some of these technologies, that we could start to subdivide and maybe grow these in individual wells or something, and then sample what is coming out and be able to make that link. But no, we do quite a bit in not only the fecal matter, but also we have some where they've grown some of the colonies up from that material, and then we do analysis on the supernatant. And we get hundreds of things. Now, the one issue I will say is we have to then have a pretty good understanding of what the beginning culture media is. Because
what did they eat to produce those? Yeah.
Because those are now going to go up and down, and it is a complex environment. It's not quite as simple as everybody would think, because at some point there are going to be markers that are going to say that it ran out of nutrients before you knew it ran out of nutrients, and they're going to give off the things to say that, and that's going to affect your results at the end. But I mean, I think we're getting better at knowing what those markers are, and as long as we incorporate those, then we get better interpretations out at the end.
No, we have not. Actually, our protocols normally enrich for the cell pellet, and then from that.
Thank you.
just to add, I think, we don't necessarily look for things that are secreted, but metatranscriptomics at least can inform what genes are being expressed and perhaps what is potentially being secreted into the extracellular matrix.
So to follow up on that, and when do your collaborators or researchers decide to use metatranscriptomics over traditional metagenomics? Like, what does metatranscriptomics tell them that metagenomics did not? I mean, of course, there is expressed genes, but
Right.
what's the follow-up after that? Do they use it for metabolomics research, or do they use it for?
It's a black box to us, unfortunately. I think they use it for more hypothesis-driven research downstream, but they don't come back and talk with us that much about what they're doing downstream of the metagenomics and metatranscriptomics. Sometimes we wish we knew, but unfortunately not. But being involved with this panel, it would be cool if they were, taking it forward. And, something that I thought of to ask and related back to the question that you asked earlier was, could the researchers be taking their metatranscriptomics data and using that to inform decisions for what type of metabolites to analyze downstream? And I don't know how much metabolomics costs these days, but I know how much I spent 15 years ago as a grad student on a big metabolomics project, and it was a pretty penny. And I don't know if targeted approaches are less than a more general approach.
I would say, generally speaking, the targeted approaches are usually more expensive than untargeted.
Okay.
So, but you get a different level of answer.
Gotcha.
I mean, they both give you peak areas, and whether or not you structure with standards and you get amount of absolute quantitation. Yeah, there's pluses and minuses. But to your point, I mean, I've had a project that just came in the door, and we just finished step one. And I say it's step one because as soon as we get back the results, they go back to their RNA-seq data, which they had, that drove us to why we did a targeted or semi-targeted assay in the first place. Then they go, oh, we're also now interested in this pathway. And then I have to say, oh, well, that would have been nice to know when we didn't run that assay. So now we have to go run another assay to capture, in this case, they were interested in the xanthine pathway, which we did a polar assay, and I don't think many of those, most of them, and we can detect some of them, but they're all in the solvent front, and I don't trust them, so they're going to have to do more analyses.
Gustavo, kind of, following up on the metatranscriptomics question I had earlier, in the field of single organism, especially when, researchers are trying to find out any variant peptides or proteins, they use RNA-seq data, generate a protein database, which has got both, known proteins as well as variant proteins. Have you or anybody in the field have they actually thought about using metatranscriptomics data to generate a database that is more customized for your sample, or do you think it's too much? It's a lot more expensive to do it that way rather than using the methods you're using right now.
Yeah, not that I know, not in our experience. Yeah, but yeah, just I think that would make the analysis, more directed. That will help a lot with the identification because the search space is a big problem in proteomics. Yeah, but.
And so most of the samples that you handle are soil and gut microbiome samples right now? Is that?
Yes.
Okay. Okay. Any more questions from the audience? All right. If not, let us thank our speakers for excellent talks and introduction to various methods that I've used in microbiome research. And I guess we can move on to our lunch. Thank you very much, everybody.